Good. Ladies and gentlemen, it's a great honor for me to be invited to participate in this meeting dedicated to John Grisham, who was an outstanding scientist and my good friend for many years. Well, and now let me turn to this the title of my presentation, and I just remind you that dopamine is considered to be synthesized in dopaminergic neurons, and as a neurotransmitter it is involved in the regulation of most functions, like cognitive functions, like neuroendocrine functions, like movement, motor behavior, etc. Almost all of the functions which we know. Okay, and so far it is believed that dopamine is synthesized only in dopaminergic neurons. But I will convince you, I hope, during my lecture, that dopamine is also synthesized by non-dopaminergic neurons, and this is among the most principal mechanisms of neuroplasticity at neurodegenerative diseases, in particular in Parkinson's disease. Well, just remind you that so-called nigrostriatal dopaminergic neurons system is involved in the regulation of motor behavior, and the cell bodies are located in the substantia nigra, and their actions are projected to the striatum. And therefore, under a loss of dopamine in the striatum, there is the Parkinson's disease is developed. Okay, what is the most important characteristics of all the neurodegenerative diseases, including Parkinson's disease? That these diseases are developed for many years, for decades, without clinical symptoms. And first symptoms appear, in this case motor symptoms, when the regulatory system, the dopaminergic system, is almost completely destroyed, and there is a deficiency of compensatory processes for this moment. And therefore, all the attempts to cure patients, by using agonists of dopamine, of neuroprotectors, they were not successful so far. Therefore, none of patients have been cured all over the world. And one of the most interesting events is the development of compensatory processes which maintain the normal regulation of motor behaviors, as I mentioned, for decades. And therefore, among the highest priorities in neurology and in psychiatry, this is an evaluation of molecular mechanisms of neuroplasticity and those that cause motor dysfunction as potential target for pharmacotherapy. And it is not very well defined, the term neuroplasticity, but still it is quite clear, and it was formulated first by Lugar and then by our scientist, Bekhterev, that he introduced the term plasticity, considering the throughout line, then anatomofunctional relations between neurons can be changed in an adaptive fashion to enable psychic maturation, learning, and even functional recovery after brain damage. Almost the same, almost in the same way, Bekhterev defined also neuroplasticity. And finally, I repeat again that during this period of preclinical stage of Parkinson's disease, it is believed that compensatory mechanisms are involved. And all people consider that these are most related to the maintaining of neurotransmission at the normal level by stimulation of neurotransmitter secretion, of dopamine secretion, by survived dopaminergic neurons, by reduction of the enzymatic degradation of neurotransmitters like monoamine oxidases, and the increase of the sensitivity of the target neurons. It means that these target neurons could react in this situation to a lower level of neurotransmitters in the extracellular space. Well, and for the study of the mechanism of neuroplasticity, particularly at the preclinical stage, and the triggers which initiate motor behavior, we have developed in our laboratory experimental models of preclinical stage and of the early clinical stage at using neurotoxin MPPP, which is injected to animals systemically. And therefore, you can say that at the preclinical stage of Parkinson's disease, although there is a depletion of dopamine, but it does not reach the threshold level of a loss of 70% of dopamine for the appearance of motor symptoms. Okay, and also at the clinical stage, the depletion just reaches this threshold level, and it provokes motor disorders. But first, we were very interested in understanding to what extent our experimental models coincide with the real pathology in human being. And therefore, we, with our collaborators, made a transcriptome of our model, and we compared the data with the data which were obtained in human beings, in patients. And you can see here that there is a striking resemblance of gene expression in patients and in mice and symptomatic mice for the most important proteins, alpha-synuclein, which is considered as a trigger of the development of Parkinson's disease, and also for most vesicle cycle-related proteins. Well, and now we use these models for a wide range of problems: evaluation of molecular mechanism, of neurodegeneration, of neuroplasticity, for a search of triggers, for a transition from preclinical stage to the clinic, for a search for peripheral biomarkers, and for the development of provocative tests for the diagnosis and the development of preventive therapy. But today we will speak about the mechanism, mostly about the mechanism of neuroplasticity. And first, certainly, we try to recognize to what extent survived dopaminergic neurons after the treatment with neurotoxin can maintain the neurotransmission at the normal level, and what might be the mechanism of neuroplasticity, which are related to the increase of the functional activity of these neurons by means of dopamine synthesis and dopamine release in the striatum. And please look here. Well, so early we have found that by means of dopamine synthesis, there is a high compensatory activity of tyrosine hydroxylase activity, gene expression, and therefore synthesis of dopamine. Well, so that is the most important compensatory mechanism. At the same time, there is another compensatory mechanism which is switched on. This is dopamine release, spontaneous and stimulated. But finally, at the symptomatic stage, the release of dopamine decreased and the dopamine uptake increased. And therefore, it causes the decrease of the concentration of extracellular dopamine, and therefore, these events could be considered as triggers for for the appearance of movement disorders. But as I promised, we will evaluate today also, are dopaminergic neurons the only one source of dopamine in the brain? Let me remind you that almost 25 years ago, after the development of double immunoleveling technique, it has been demonstrated that in addition to dopaminergic neurons, which co-express both enzymes of dopamine synthesis, tyrosine hydroxylase and aromatic amino acid decarboxylase, people found non-dopaminergic neurons which only partly express dopaminergic phenotype and only individual enzymes of dopamine synthesis, either tyrosine hydroxylase or decarboxylase. And that was very intriguing, and therefore the most brilliant people from Europe and the United States, they joined their facilities and intellectual potential to understand what is the functional significance of these neurons. And unfortunately, they failed to do it. And we have returned to this issue only at the beginning of 2000 by demonstrating that these so-called monoenzymatic neurons are widely distributed all through the brain, and the number of these neurons highly exceeds that of true dopaminergic neurons. And we were very lucky when studying the development of the hypothalamic neuroendocrine system, we found that at that stage, only at that stage, the arcuate nucleus, which is considered as a dopaminergic center in adult animals, well, so at the end of fetal life, this arcuate nucleus consists only presumably of monoenzymatic neurons, for more than 99% of monoenzymatic neurons. And despite an absence of dopaminergic neurons, there is a high level of dopamine synthesis that has been demonstrated both in vivo and in vitro. And according to our hypothesis, this dopamine is synthesized by monoenzymatic neurons in cooperation. It means that L-dopa, which is produced in Th neurons, is transferred to the decarboxylase containing neurons for the final synthesis of dopamine. In order to test this hypothesis, we inhibited the so-called semi-specific transporter, membrane transporter for L-dopa for other neutral amino acids with L-tyrosine. And look, please, that in this case, the dopamine synthesis dropped in the arcuate nucleus, which was enriched by monoenzymatic neurons, but it was increased in the substantia nigra, which contains only dopaminergic neurons. It is naturally because tyrosine is a substrate for dopamine synthesis. Well, and then we have demonstrated that these neurons are characterized by very intimate, very close relations, which serve at the electron microscopic level, at the confocal level, which serves to most efficient transfer of L-dopa from one cell to another. This is a schematic representation of dopamine synthesis in dopaminergic neurons, and this one in monoenzymatic neurons. This is cooperative synthesis in monoenzymatic neurons. Okay. And then the next question was, at studying the hypothalamus, what might be the functional significance of this cooperative synthesis? And we have remodeled the neurodegenerative disease, which is called hyperprolactinemia. It means that dopamine, which is produced in the arcuate nucleus, it inhibits the secretion of prolactin. And therefore, when we provoked the degradation of 50% of dopaminergic in the arcuate nucleus, first, we came to the increased secretion of prolactin. But sometimes later, there was a return, the prolactin secretion to a normal level. And this was also the case for dopamine synthesis. Dopamine synthesis was increased until the normal level because the number of monoenzymatic neurons increased considerably. And that was the first evidence that this is a compensatory reaction at the degeneration of dopaminergic neurons. And now the last question, are there monoenzymatic neurons in the striatum of normal and Parkinsonian mice? And this is the most important mechanism of the development of Parkinson's disease. Well, and in fact, we have demonstrated that we were unable to find monoenzymatic neurons in the striatum of intact animals. But after the dopaminergic neurons degeneration, we have found monoenzymatic TH neurons, TH-containing neurons in the striatum, and at the same time, the gene expression was highly increased in this particular region of the brain. These are the evidence that monoenzymatic neurons appear in the striatum, but only under the degeneration of dopaminergic neurons. But in contrast to dopaminergic neurons, which are few in size, the number of monoenzymatic fibers was strikingly large, and therefore we have demonstrated also that under degeneration of dopaminergic neurons and differentiation of the striatum, there is an increase, a progressive increase of the nerve fibers which contain only one enzyme of dopamine synthesis. You know certainly that dopamine synthesis is cytosolic, and therefore it is synthesized not only in cell bodies but also in the axons. Well, and finally, we tried to to find out are there other sources of monoaminergic fibers in the striatum except the intrinsic neurons. And when using the axonal the marker of axonal transport, we have demonstrated that there are, outside the striatum, the monoenzymatic neurons which project their actions to the striatum, but only few neurons are there. Well, so that's not important. And then for coming to the direct evidence of dopamine synthesis by monoenzymatic neurons, we have used the same methodology that was used already for the hypothalamus. Well, so we inhibited the A-dopa transport of the decarboxylase cells by using L-leucine or histidine. These are amino acids which are completely indifferent to dopamine synthesis. And in this case, we came to the observation, we observed that the L-dopa concentration in the extracellular space increased considerably. That is, naturally, we prevented its penetration to decarboxylase cells. And the dopamine synthesis in monoenzymatic neurons decreased progressively as the differentiation of dopaminergic neurons proceeds. But in the same experiment, with the same design of the experiment, we didn't find any difference in dopamine synthesis in the substantia nigra, which contains only dopaminergic neurons. And the last question was, what might be the mechanism, the molecular mechanism of the regulation of expression of the enzymes, of the individual enzymes of dopamine synthesis in non-dopaminergic neurons. And you can see that the number of TH cells in the striatum decreases progressively in ontogenesis just under the establishment of afferent innervation of these neurons. And therefore we suggested that the afferent innervation catecholaminagic fibers inhibit the tyrosine hydroxylase expression. And we have proved this by using vasopressinergic neurons which co-express tyrosine hydroxylase, but when pharmacologically cutting the influence of noradrenaline. Well, and now let's come to the conclusions. The striatum of normal and MPTP-treated mice contains monoenzymatic neurons expressing individual enzymes of dopamine synthesis. Monoenzymatic neurons expressing complementary enzymes, tyrosine hydroxylase or aromatic amino acid decarboxylase, synthesize dopamine in cooperation, but only in Parkinsonian mice. Synthesis of dopamine by the striatum monoenzymatic neurons is enhanced as dopaminergic neurons degenerate. Well, so we avoid of catecholaminagic inhibitor influence of tyrosine hydroxylase expression. And at last, synthesis of dopamine by monoenzymatic neurons appears to be among the most efficient mechanisms of neuroplasticity at neurodegenerative diseases, including Parkinson's disease. Well, so I use this opportunity to express my acknowledgement to my staff. These are very talented and enthusiastic people, and finally, so having this presentation and assisting at the introduction, I have a feeling that that Jenny still is with us, and he enjoys this meeting. Thanks for your time.
Thank you very much for this highly significant and original presentation. I think we steal a couple of minutes from the next speakers, allowing a couple of short questions, short answers.
Thank you for interesting talk. And do you see any perspectives to increase the production of dopamine through these non-dopaminergic neurons, probably adding something, acting on them?
I think that this is a potential target for neuroprotective therapy, which is developed all over the world and also in our country. And it is particularly important for monkeys and for patients, because the number of these monoenzymatic neurons in the striatum of these patients is multiply higher than the number of these neurons in rodents. Well, so I think that is a very, very potentially interesting target.
My question was, do you see the way to reach those non-dopamine neurons today, or there are no way yet, no ways yet?
Today. We should recognize what are the mechanisms of the control of the expression of these individual enzymes, actually in non-dopaminergic neurons. Well, for the moment we know only one, that this expression is under the inhibitory control by catecholamines. But still, I think we should learn all the mechanisms of the regulation at the molecular level, and after all, we could come to the pharmacotherapy, I think. But this is, I think, a rather long way.
Thank you very much. I hand over to my co-chair, Professor Vadim Ivanov.